I'm Wendy Silk, talking to you at the 11th Symposium of the International Society of Root Research and Rooting 2021. During this difficult year of the COVID pandemic, with all its social isolation, we can still communicate across vast distances around the globe. For this wonderful opportunity, I thank the organizers and the internet. I'm going to be talking about moving with the flow, root growth, and our changing environment. You may wonder what roots have to do with flow. I hope by the end of this talk, I'll convince you that approaches from fluid dynamics are powerful tools to understand root development, how roots respond to environmental change, and how roots in turn affect the soil. I like to introduce this topic by talking about the growth not of a root, but of a hypocotyl. And those of us who are gardeners know that dicot seedlings sprout, they come out of the soil with a little hook. This hook is thought to be maintained during the growth of the seedling through the soil. It's thought to protect the important cells of the apical meristem from abrasion as the seedling pokes up through the soil. And then light acts as a signal to flip it open once it emerges, and it will then resume its normal straight growth trajectory. So I want to show you a movie in which the hypocotyl is maintained as a hook shape in green light, which causes it to remain etiolated. So here we have this poor seedling growing in a scintillation vial in green light, which cause it to stay etiolated and maintain its hook. And I want you to watch what happens to a mark on the surface. And as the plant grows, you'll see the hook stays about a centimeter back from the apical meristem. Here are the cotyledons. Meristem is right here between them. Oh, look, our mark has now gotten to the hook summit, but it doesn't stop. It's going to flow through the hook, and eventually we'll see it on the basal side of the hook. And now look, it's almost straight, and now in fact it's part of tissue that's straight. And we can reverse this process using the miracle of modern time-lapse photography, and we see once again our mark is on the summit of the hook, and we'll push it back even further in time and we'll find it in its initial location on the straight apical portion. Now we know that plant tissue, unlike animal tissue, doesn't race around during development. It's all cemented together, and it's just pushed by the and stretched by expansion of its own cells and adjacent cells. So what this movie tells us is that the hook, which is maintained about a centimeter back from the tip, is actually produced by a parade of tissue elements, each of which first curves and then straightens. And it's this parade of changing elements that produces the form that, at least from the tip of the plant, looks fairly constant. I'll be talking about plant growth kinematics. This is a term used in fluid dynamics to refer to movement or changing shape before you get to discussing the underlying mechanism. Watching the growing hypocotyl hook was a good way to see the analogy between plant growth and fluid flow. But hypocotyls aren't the only plant structures that resemble fluids. In fact, all of the growing parts of plants, in particular the growth zones of roots, can be analyzed in these terms. Both roots and fluid structures such as rivers and fountains are structures that are made of changing elements, and in the case of roots we have the fundamental element of the cell, whereas in a river or a fountain the moving element is a water droplet. But in both these structures, the structure is formed by a parade of elements which moves through the structure and experiences continuing change. So an important concept is the distinction between a material and a spatial description of growth. Not just growth, but any developmental variable that can be specified in terms of position in the plant and time. We could have a spatial description. The spatial specification considers the variable at particular locations in the plant at a single instant in time. So in the literature, for instance, we often see plots of enzyme activity or organelle size. We can talk about cell division rates that are high in the region 1.5 millimeters from the apex, and they are much lower or perhaps 0 once we get to be 5 millimeters back from the root tip, and so forth. A different way to look at the root, the growing root, is to have a material description or material specification where we're going to follow a real or material element of the tissue, such as a cell wall. And with time, that element is bound progressively farther from the apex. So for a given cell cohort, our material element, in this example, the division rate is high while the cell is in the meristem, and then the division rate drops to 0 when the cells are displaced past the meristem boundary. And to understand the plant, we need to appreciate both the spatial and the material aspects of the development. Let's just think back to that movie of the seedling growth. If we're interested in understanding the mechanism of hook maintenance, now from the time of Darwin, whenever a botanist sees a curved form, they look for a growth promoter to be more abundant on the outside than the inside of the form, because we know that the outside is longer and therefore it must have had more growth. I would argue that if we found a growth promoter that was more abundant outside than on the inside of the hook, we would have proved conclusively that this was not responsible for hook maintenance. Because to understand the hook, we have to appreciate that here on the apical side of the summit, the elements are increasing in curvature. So yes, here there must be a growth promoter on the outside of the hook, or possibly a growth inhibitor on the inside. But once we pass the cell summit, the element is straightening. So here on the other side, the basal side of the summit, we need to have a growth promoter more abundant on the inside, or a growth inhibitor on the outside. And it's only by thinking not only of the property of the tissue where it is instantaneously located, but also what its fate will be, that we can begin to understand the mechanism of the pattern that we're looking at. Another important concept is the distinction between a moving and a stationary reference frame. And for this, I like to use the analogy to a boat wake. If we're in a boat looking back, we see the wake, and if the boat is moving at a constant rate and not changing direction, we may see a pattern of the boat wake that isn't changing in time. 10 feet back from the boat, we see a particular pattern, and then 20 feet back, we see a different pattern, and so forth. However, if we stand on the shore and look at that same pattern, we see something that's changing constantly. And we appreciate then that the steady pattern, that is the pattern which isn't changing in time, is evident only in a moving reference frame. So we have to stand in the boat, sit in the boat, or sit on the tip of a root to see the pattern that is unchanging in time or slowly changing in time. So, of course, this is the reference frame that is instinctively chosen by the anatomist. And in any elementary botany textbook, you'll see a micrograph or perhaps a diagram of the root tip. And there'll be perhaps no mention of the age of the root you're looking at. It won't even mention at first that this whole pattern is moving down through the soil. You'll see the root cap, which is in fact a separate little organ with its own time scale for turnover. And then the textbook will show that cells are dividing and expanding close to the tip, and further back they continue to expand for some distance. Phloem differentiates in the distal region of the apical meristem. Further back, you become aware that the xylem cells are differentiating. Beyond the frame of this micrograph, we know that at maturity these xylem cells will be dead, very thick-walled, specialized for carrying water. Also, just beyond this micrograph frame, we know the root hairs will differentiate and so forth. So the anatomist sits on the tip and looks back to see this pattern. The root growth zone may be steady if we look back from the tip, but it's only in this micrograph that the cells are fixed. In a living root, the cells are displaced through the standing pattern, so that the cells which are dividing now will, in a few hours, cease dividing. They'll start to make xylem when they're a few millimeters back. When they are 6 millimeters back, an hour or so later, they'll start to make root hairs, and so forth. And the agronomist will be aware of another aspect of the root development, which is not evident in the micrograph, and that is that the root tip will be moving down through the soil. And some of you may have been mesmerized by this video here of growth of a marked root. And you'll see that the tip is just inexorably pushed down through the soil by the growth of the cells behind the tip. It's important to recognize that there are spatial and material aspects of growth itself. And here I show 3 figures that illustrate the analysis of growth of a corn root. The central figure here is a plot of the displacement velocity. We put marks on the root, and you do a fairly short-term analysis of the movement of the marks, and then you calculate the rate of displacement from the tip, and you can plot that versus distance from the tip. And it was Ralph Erickson who showed that the derivative of velocity with respect to position gives a relative growth rate at a point on the root. And he called this the relative elemental growth rate because it's the relative growth rate of a tiny element of tissue. And when you take the derivative of growth velocity with respect to position, you find that growth is about at a maximum of 40% per hour at 2 millimeters back from the tip till about 7-8 millimeters back, and then growth has ceased by 11 millimeters behind the tip. So these 2 figures, the growth velocity curve and the relative elemental growth rate profile, these are spatial descriptions of growth. We're looking at all of the particles on the root at a single instant in time. There's another way of characterizing growth, perhaps an even more fundamental way, and that is to follow a particular real or material element, perhaps a cell wall, as it moves during root growth. And you see that with time, our cell wall is found further and further back from the tip. In fact, it accelerates away from the tip until it reaches this constant velocity equal to the root elongation rate. This happens when it's out of the growth zone. And in fact, there's a whole family of what I call the growth trajectory, depending on which particle you choose to start following and where it's located. Now, this is a material description of growth because we're following the actual cell wall or other particle that is a real or material element of the structure. For any of you working on growing tissue, I urge you to do a growth analysis, do a marking experiment, work up the growth velocity profile, and calculate the relative elemental growth rate pattern and work up the growth trajectory. There'll be many uses for these numbers, as I hope to show later in the talk. So here are some collaborators from my early work in plant growth kinematics. It's the wonderful thing about science is that it's most often a team sport, and I've had the wonderful privilege of working with people from all over the world with a suite of all kinds of talents. My great mentor, Ralph Erickson, was the first to do a quantitative analysis of root growth. His work is my frame of reference. When I got to Davis, I asked Ted Schaut to be my co-investigator on my first NSF project, and we were lucky enough to attract a brilliant young postdoc. Bob Sharp came to us from John Boyer's lab, and Bob went on to become more distinguished than either of us. He's done groundbreaking work on plant response to drought, and he's rewritten the textbook on hormone interactions during development. When we're talking about roots, there are at least three different points of view that we need to be able to take. We want to look from the point of view of the moving root tip, look out. Then we want to take the point of view of a soil scientist. The soil scientist stands on the soil surface and looks down and sees the arrangement of the soil horizons in a particular order. And so we need to be able to be comfortable in this frame of reference. And then from the description of kinematics, we recognize that we want to be able to do a material description of the growth too. We want to be a particle initially attached to the root tip and located in time further back from the tip as growth occurs. So from the point of view of the moving root tip, we've seen that root anatomy is best described. It's also a good way to see the environmental effects on a root growth rate pattern. And then looking out from the root tip, we can talk about the chemical fields that surround the tip and are important in understanding how the root affects its own environment. And I'll give you an example of a nutritional study, nitrogen nutrition in the growth zone. From the point of view of the stationary soil horizon, we can see development of the rhizosphere. and we can see root strengthening of soil. And then from the point of view of the particle that's attached to the root tip, we'll see a growth trajectory in a moving and also in a stationary reference frame. And it's the way we need to look at bacteria in the rhizosphere of the tip. So for the next part of the talk, I'll be giving a hop, skip, and jump through some of my favorite papers that will illustrate these different points of view. So we want to get comfortable jumping on and off the root tip. What is the connection between the moving and the stationary frame? In the moving frame, we have distance x measured from the tip. In the stationary frame, we have distance z measured down from a stationary soil particle. And we see that the relationship is pretty simple. X plus z equals the length of the root plus some arbitrary constant. And a corollary is that the velocity of displacement from the tip plus the velocity with which our root particle is moving away from a particle in the soil is equal to the elongation rate of the root. So this was the transformation that Ralph Erickson used in his classic root growth analysis, and we'll see it again in analyses of pH in the rhizosphere and bacterial abundance in the rhizosphere of the root tip. So what do we see when we look out from the root apex? This is an excellent reference frame to look at the impact of environmental variation on growth. And for instance, if we do a growth analysis for a temperature perturbation and a change in water stress, both of which reduce the elongation rate to perhaps a quarter of a control value, we see completely different spatial patterns. And this tells us that the mechanism of response to these two environmental variables must be completely different. Then to push back to the molecular basis of the regulation of the growth rate, we need to look at the abundance of a gene or a message and compare that to the spatial profile in the relative elemental growth rate. And this kind of work is going on around the world in Bob Sharp's lab here in Missouri, for instance, in Beatrice Tribulot's lab and Gerrit Beemster's lab in Europe. And kinematic analysis is continuing to illuminate how plants resist drought in particular. Now, I'd like to tell you about some nutritional studies, but before I do that, I'll have to step back and show how we need growth analysis also to calculate a biosynthesis rate in a growing tissue. I use a form of a continuity equation. So what we want to calculate eventually is the deposition rate and its spatial pattern, where the deposition rate is a production or a degradation rate, perhaps in micrograms per millimeter of root. So the variables that we're looking at are the concentration of the metabolites, such as protein, given in micrograms per millimeter, time, which for a root study is typically measured in hours, and our old friend, the growth velocity, and our old friend, distance from the tip. And the continuity equation tells us that there are three different components to consider and to calculate. This first change, the local rate of change, that's all the biochemist usually thinks about. He has a well-stirred beaker and a reagent, perhaps a colorimetric assay for protein, where the more blue we have in the beaker, the more protein there is. So if he finds more blue, that's the protein production. And if he finds the blue color is fading, then he knows the protein is being degraded. And it's very simple then to calculate the production rate or the degradation rate of protein. But in a growing root along the tip, we may see for hours or even days that there's no change in the spatial profile of the concentration. That doesn't mean the root isn't making protein. Of course it's making protein, it's growing. And this term here, the growth dilution term, is pretty intuitive actually. Remember, we found that the root was growing 40% per hour. So if at that location we find 2 micrograms per millimeter of protein, we know that 40% of that concentration must be added each hour to maintain the concentration pattern. Then this third term, to me that wasn't so intuitive. It's called the convective rate of change. It's the product of growth velocity and spatial gradient in protein concentration. I find it hard to understand until I thought of a physical analogy. Let's imagine that there's a school of fishes swimming in a stream,
And we're an artist and we'd like to capture this pattern. How can we keep the pattern still long enough to paint it? Well, that's a riddle. Those fish are just moving along. Well, the answer, of course, is we have to hire workers to throw fish into the stream, so that when this column of 5 fishes disappears under the bridge, this worker will throw a fish onto the column of 4. This worker will throw a fish onto the column of 3. And now we see that, yes, the faster they're moving, well, the faster those workers have to be throwing their fishes. And also, the greater the difference in the number of fish from one location to the next, the faster the workers will need to be throwing their fish. So, starting to believe the convective rate of change, then, we go back to our full equation, and we see that to calculate the spatial pattern of protein deposition, we'll be looking at the spatial pattern of protein along the route, and we'll have to look at it at 1 or 2 times to see whether or not it's steady, what the local rate of change is. And then we'll run up these products involving the relative elemental growth rate, the growth velocity, and the local protein concentration, and we will be able then to calculate the rate at which protein is being produced. So I'll just give you an example of nitrogen nutrition in the root growth zone. Arnold Bloom has built microelectrodes to measure the flux of both ammonium and nitrate into the root. So we had the spatial pattern of flux along the tip. We did a growth analysis, and then we measured the ammonium and nitrate concentration profiles to go along with that data, plugged it into the continuity equation, and found eventually, when we put it all together, that in the meristem, the deposition of ammonium exceeded the influx. So here, the tissue was a local sink for ammonium. It was importing the ammonium. Further back, the ammonium influx was greater than the deposition rate, and so the tissue was exporting the substance. It was a source. For nitrate, the influx all up and down the growth zone was greater than the local deposition rate. So for nitrate, the root was everywhere doing its traditional function of supplying nitrate to the shoot, as well as the root. And we made the interesting discovery that the deposition rate in the basal part of the growth zone was high enough that nitrate was a significant part of the osmotic pool there, sustaining the rapid growth rate. So that was kind of an interesting conclusion as well. So I've done, over the years, a number of nutritional studies illuminating plant response to heavy metal stress, to salinity stress, to drought stress. Collaborated in particular in teaching with my Davis colleague, Andre Loikley. We taught for many years plant response to environmental stress. So this approach, combining a formulation from fluid dynamics with plant growth analysis and traditional plant biochemistry, has provided a number of insights over the years. Let's turn now to the mechanics of root growth. The primary growth process is water uptake that inflates the cell, and the cell expansion is sustained by deposition of metabolites and wall components. So a fundamental question for root growth mechanics is, what is the spatial pattern of water potential that sustains an observed growth pattern? So we have to modify Darcy's fundamental law, because only water that's moving faster than the cell wall is moving will cross the wall to inflate the cell. And we find eventually that the growth rate in 3 dimensions is related to the hydraulic conductivity tensor dotted into the water potential gradient. And we further observe that there's no functional water conducting element in the root growth zone. Water for growth must come entirely from the soil, and possibly, preview of coming attractions, from photofloem. So here is an analysis of growth sustaining water potentials, work done with my colleague Angela Cheer and our student Brandy Wiegers. And we found that the only boundary conditions that gave reasonable results had water coming from phloem and soil. And that's these results here. Only these results were consistent with what experimentalists were finding with miniaturized pressure probes and psychrometers. So here we have water supplied by the phloem and also by the soil. Our sensitivity analysis showed that an important parameter was how close the phloem differentiated to the tip. And it became progressively easier for the plant to grow, that it was possible to have an observed growth pattern with a smaller gradient in water potential if the phloem differentiated closer to the tip. So a conclusion was that adaptation to drought and other environmental stress should involve more apical differentiation of phloem and or higher phloem delivery rates to the growth zone. Now, of course, the water comes ultimately from the soil into the mature part of the root where the xylem is functional, but some of it must be resupplied to the growth zone via the phloem. And we got some empirical verification of phloem water delivery. John Boyer was working with Michelle Watt in her laboratory on his sabbatical, and they invited me to kibitz on this project, and of course I was delighted to do that. They were using this time-lapse photography device that I had left in Michelle's lab when I was on sabbatical there, and John modified it so that he could look at the growth of root in both moist soil and in air, and then he snipped off the top of the plants so that phloem delivery would be stopped. He found that very soon after excision, the roots stopped growing in air. In soil, they did continue to grow, and we were able to calculate that somewhere between 1/4 and 2/3 of the water for growth was supplied by the phloem. The soil was providing the rest. Now let's look out from the root tip and ask what the soil looks like. We're still in the moving reference frame, but we're going to look out from the root to the soil. And this leads to the question of rhizosphere pH, the core question in plant nutrition. And the axiom is that the plant has to balance electrical charge when it takes up nutritional ions from the soil. To balance the charge, the plant will release or take up protons or bicarbonate ions. And this changes the acidity of the soil next to the root, the rhizosphere. Rhizosphere pH, in turn, affects both nutrient uptake and the uptake of heavy metals. We had a growth diffusion model, and I was working again with Angela Cheer and our students Tom Kim and Sue Nickel. We started with Nye's model. Nye modeled rhizosphere pH in the mature part of the root as diffusion from a cylindrical surface into a soil with a particular diffusivity. So it was a diffusion problem. We took growth into account, and we had to use the moving reference frame attached to the root tip. So here's our old friend, the coordinate transformation. And we could see that soil pH is going to be related not just to proton diffusivity, but also to the elongation rate. And in fact, the ratio of elongation rate to diffusivity determines the shape of the rhizosphere. So we came up with a dimensionless number equal to the elongation rate times the characteristic length of the growth zone divided by soil diffusivity. And this would tell us about the timescale of diffusion compared to the timescale for growth. And with the model, we could see that the rhizosphere in soil would soon be much smaller than it would be in water or agar. Here is the prediction for rhizosphere size from the model. After 12 hours in growth, the root should acidify the soil to about 1 millimeter away from its surface, whereas in water or agar, the root tip should be surrounded by an acidified layer of about 1 centimeter after just 2 hours of growth. And moreover, the pattern quickly became steady after 24 hours. And we were able to, this wasn't just a model, Sue Nickel got in there with microprobes for pH measurement, and she was able to verify these predictions. Also, later in years, there were some beautiful studies in the literature with planar optodes, and we could see the patterns there as well. So the implication is that the root tip carries a chemical field as it moves through the soil. And since the root has developmental gradients in uptake and efflux, the rhizosphere is going to have spatial variation along the root axis. And now we're ready to hop off the root and take the point of view of a soil particle. And to take this approach, I flew to Australia to work with Michelle Watt and John Paciora. I met Michelle at an ISRR meeting in Nagoya in 2001. I knew that of all the people in the world, she would be the best one to consider this problem. So let's look at a soil particle down here. See this little white square? And right now, it's probably not being influenced very much by the root. It's pretty far away for any kind of diffusion to be happening. It hasn't yet been born into the rhizosphere. But as the root grows, it may soon be receiving mucilage from the root cap. And now, oh look, it's probably getting border cells sloughed off also from the cap. Oh, now it's probably getting exudates from the growth zone. And now it's almost certainly feeling the suction of the transpiration stream. It's next to the part of the root that has differentiated xylem, so it'll be losing water. And we see that our soil particle is next to progressively older parts of the root. And this leads to the recognition that there are several different kinds of relationship with the growing root. If a soil particle is not attached, it will at first be next to the root cap. Eventually it'll be in the growth zone. Later it'll be next to root hairs, and so forth. On the other hand, an insect that's capable of moving to keep up with the zone may be able to stay on the tip where it likes the exudates, or may shelter in the root hairs and move down as they are progressively formed. For a bacterium that attaches somewhere near the root tip, there'll be a different sort of relationship. The particle that attaches will accelerate away from the root tip at the same time it's moving down through the soil. And this leads to the notion of rhizosphere development. We see here a time-lapse record of a marked seedling root. If we're on a soil particle here and we ask, when did this particle first start to be influenced by the root, we look back and we see, oh, it was 6 hours ago that it first encountered the root tip. And this leads to the idea that the rhizosphere is a chronosequence. Here's an application to soil strength. Roots are well known to increase the shear strength of soils. Planted soils are more resistant to landslides and to erosion. The factors that are usually considered for root stabilization of soils are the tensile strength of the root and the root density in the soil, usually characterized by the root area ratio, the area of roots divided by the area of the soil column. And the existing models focus on this root area ratio. So we asked, what are the spatial and temporal patterns of soil strengthening factors in a planted soil? We estimated root area ratio in a conventional way by scanning excavated roots with software by WinRhizo. We characterized both tensile strength and root area ratio at 3 different stages. At 1 week, which was the establishment of the seminal roots of these Avena fatua plants. At 3 weeks, when lateral roots had grown. And at 7 weeks, when reproductive growth had occurred. And you'll see that the biggest increase in root area ratio occurred at 7 weeks in the upper layer of soil, which was most likely to slide. Then we chopped up the root into centimeter lengths, keeping track of where they were located, and we subjected them to a breaking test. This is a poor woman's instron. These are modified paper clips clamping the root. The paper clip was suspending a large plastic bottle. We introduced water into the bottle very gently until it got heavy enough to break the root, and that gave us the force which was required to break the root segment. We found a spatial gradient in root tensile strength over at least 40 centimeters, and this occurred in both moist and dry soil. So there were implications for temporal development of soil strength. The tensile strength increased spatially greater than 15 times between the tip and the 40 centimeter location, and they were growing about 1 millimeter an hour in moist soil. So therefore, strength in the soil layer, due to increasing root tensile strength, should increase for those 17 days after arrival of the root tip. So here we are in the stationary soil layer that's most apt to slip, and we're impacted by a very weak root here, and then after 17 days we have 15 megapascals of tensile strength. So we can conclude that root developmental gradients should be incorporated into complete models of soil strength. Now let's move on to the third point of view. What does a particle attached to the root tip see in the soil and see on the root? And for this question, we'll be considering growth trajectories in both moving and stationary reference frames. And this is the point of view that's relevant for many bacteria in the rhizosphere. The rhizosphere has become the wild frontier in root research. And I can quote from a recent review by Carla de la Fuente Canto and Malcolm Bennett and others. Dynamic interplay in root-soil microbiome interactions creates emerging properties that impact plant nutrition and health. The rhizosphere can be considered an extended root phenotype, a manifestation of the effects of plant genes on their environment. Here are some curves to keep in mind. The growth trajectory in the moving reference frame, which we've talked about. The bacteria attached to a root tip will accelerate away from the tip until they achieve a constant velocity equal to the root elongation rate. And at the same time, the bacteria will be moving downward in the soil more and more slowly until it is in the non-growing part of the root. So this was one of my favorite papers. It was a model of bacterial colonization of root tips by Lionel Dupuy. It was a model on a sensitivity analysis. We were inspired by the knowledge that bacteria may colonize the root tip, although some root tips are sterile. And there are challenges to attach to the root tip because we've seen the cells are moving, they're expanding, that will dilute any bacterial division. There's efflux, there's sloughing of border cells, lots of challenges. Lionel put together a model, and he has terrific math skills, and the model includes dynamic visualizations of bacterial plant interactions. So I hope you'll buttonhole him. He's here at this meeting. Even better, invite him to come talk at your institution. He has a lot to say. There are just some fascinating ramifications of this model. First of all, we have to admit that it's extremely complex. It's got elements from bacteriology, biochemistry, botany, soil science, soil physics, and that's a great challenge for working in the rhizosphere. It's so darn multidisciplinary. Well, we got some fascinating results. We came up with dimensionless parameters for chemotaxis of the bacteria and the bacterial dispersion. But the model showed that variation in chemotaxis and dispersion don't matter very much unless the root has stopped growing. So our sensitivity analysis looked at the attached bacteria profile with variation in the parameters in the model. Attachment had a slight effect. Length of the growth zone didn't have any effect on the colonization near the tip. The most important parameter by far was the elongation rate of the root. And Michelle Watt had found some empirical evidence substantiating this result as well. Hardly any effect of chemotaxis on the colonization of the tip. And another fascinating result was that for the model to work at all, the root cap has to have a carrying capacity for bacteria with a nonlinear dependence on bacterial density. This was a picture of the model. We think that colonization starts in the cap. Border cells that are sloughed can contribute to colonization further back. The bacteria remain rather sparse while they're being diluted. As growth slows, they can start to exceed the carrying capacity of the rhizoplane and to disperse to enrich the bacterial abundance in the rhizosphere just behind the growth zone. So the take-home message, no surprise, paying attention to root growth leads to an understanding of rhizosphere development and many aspects of root-soil interactions. Now I'd like to change gears and talk about science and the public. At this meeting, we are all aware of rapid climate change. And as scientists, we know that answering one question inevitably leads to posing new ones. This wonder is part of the excitement of science. So we have no trouble recognizing that the technologies and public policies that greatly benefit one generation can prove harmful in later eras. As our human population grows, we need renewable energy sources and eco-friendly products, manufacturing processes, and public policies. Science and technology are more important than ever before. How can we tell the public both the scientific approach and give them an understanding of natural history? The United States National Science Foundation is concerned about education, of course. Before 2016, I was part of a task force called Vision and Change in Undergraduate Biology Education, a call to action. And these were the important recommendations that came out of our report. Science education should involve active learning. It should be student-centered, should be experiential, stress science as a process, not just natural history. Concepts should be developed and the facts embedded in those concepts. There should be societal relevance, and we should do a better job of engaging our diverse population and communicating across disciplines. On my own campus at UC Davis, I was part of an educational effort called the Art Science Fusion Program, which at the time I joined it in 2007 was still a fairly new paradigm for experiential learning. We justified our approach by finding evidence that the process of hands-on creativity anchors learning. If you're drawing or dancing or singing, all of your senses are involved, and research shows that there's better learning. We also saw very clearly that the art activity is a strong motivational force, and it seems that when students are encouraged to be creative on their own, that they become very engaged with the subject. And the effectiveness of this approach during the time that I was involved in the program became very well demonstrated in K-12. My colleague in education, Meryl Goldberg, who works at Cal State University San Marcos, had a large-scale educational research project in which she introduced art into the curriculum in some schools that had very poor achievement test scores. And what they found was that within 2 years, these students that had been underperforming started to score above grade level on their standardized achievement tests, while students who had not had arts projects introduced into their traditional curriculum remained below grade level. My contribution to this program was to teach a course called Earth, Water, Science Song, Science and Society 42, in which students, instead of writing exams for their professor, wrote songs, and instead of having a final exam, had a final musical where they were charged with teaching the larger community what they had learned about environmental science. So on the screen here you see the flyer that went out to entice students to enroll in this class. And I'll show you next the closing number for one of the final performances in Earth, Water, Science Song.
So the pedon is the basic unit of measurement of soil. It's a meter by 1.5 meters down. And we're going to tell you about all the history that can be contained in that one small volume. So this song is called Still That Same Pedon.
1, 2, 3, 4.
Hey!
Foundation for our structures, an environment for crops, more complex than you'd expect, not just dirt and rocks. Soil contains a history that's built up all these years. Snow and wind and rain and water, erosion happens here.
Nurturing home earthworms, breaking down the dead, nutrients for the soil, making sure we're fed. We always measure soil in units called pedons, and horizontal layers called toil horizon.
A meter by another and a half, changing but persistent. We gotta make it last. No matter what happens, no matter what's gone on, the soil's always changing, but it's still that same pedon.
And on
Look at the pleasure on those student faces. We saw an increase in self-esteem, kind of unusual in a science class. So I encourage you to try assigning some arts projects in your science classes. A haiku, a rap, a sketch, a skit, a drawing, a painting. You'll see increase in student engagement and also in long-term retention of the concepts.